So welcome to the fourth and final week of the Duke-CZI Bench to Bedside Speaker Series. My name is Luke Bartelt, and along with my colleagues Pia and Shi, and with the help of the Chan Zuckerberg Initiative, we've been organizing the Neurodegeneration Challenge Network here in RTP, North Carolina. Our goal is to connect researchers, clinicians, biotech industry workers, and patients in the area to foster collaborations and navigate the barriers to push forward neurodegenerative research to work towards better therapies and cures for the patients here and around the world. We're really excited today for our speaker, Dr. Kurt Fischbeck. But before I introduce him, I just want to go over some logistics. For anyone who has not attended some of the previous weeks, what we're doing here is a talk, which will be followed by a Q&A session. So if you have any questions throughout the talk, you can drop them in the chat and I'll get to them during that session. And then after that Q&A, we'll go into some breakout groups to get some more in-depth discussion about the topics that we're going to be covering today. So without further ado, Dr. Fischbeck has been a central contributor to the neurodegeneration research field for nearly 40 years. Kurt earned his bachelor's and master's degrees from Harvard University in 1972 and his MD from Johns Hopkins in 1976. After completing a neurology residency at UC San Francisco, Kurt moved into academic research as a postdoc working on muscular dystrophy at the University of Pennsylvania. In 1982, Kurt joined the faculty there and started his own research lab at UPenn and immediately began making significant contributions to muscular dystrophy research. He quickly expanded into several other diseases, tracking down and mapping the genetic loci for heritable diseases, and discovering that it was a polyglutamine expansion that was responsible for spinal bulbar muscular atrophy. Since that discovery, Kurt's research has dug into the mechanisms of these diseases and has taken him all the way into clinical trials for new and promising therapies. I'm sure it's a great joy to be involved in research of a disease end-to-end like that. And it's for that reason that I am extremely excited to hear his perspective today on the role of academic researchers in pushing forward cures for neurodegenerative diseases. So without further ado, please join me in giving a big virtual round of applause for Dr. Kurt Fischbeck. Take it away, Kurt.
Thank you very much. I'll go ahead with the share screen here. Let's see if this works again. Can everybody see that okay? Yep, nodding. Okay, great. And you can hear me okay too. Well, I'm really pleased to be a part of this series and kind of a closer for the series. It's a great time to be talking about development of treatment for hereditary neurologic diseases. We've been working, as Luke was saying, we've been working in this field for a long time, and it's finally getting to the point where we're seeing some successes. And so it's nice to highlight those and hopefully inspire some of you all to pick up the gauntlet and carry the field further, because it's clear that there's a lot more work to be done too for a lot of diseases. So before I get into it, I just wanted to say something about the outside advisory work that I do, consulting work that I do. I am a government employee, so head up the Neurogenetics Branch here in the intramural NIH in Bethesda. So we have a group of about 7 different investigators, and maybe including all the trainees and support personnel, about 90 people in the branch. But I also do advisory work for other organizations. And as a government employee, I can't get paid for doing outside work, but that doesn't seem to prevent people from asking me to do advising work. And that includes nonprofits like the Muscular Dystrophy Association, the French Muscular Dystrophy Association, and disease-specific groups like the Spinal Muscular Atrophy Foundation. and target ALS. And this is part of a larger list that relates to this particular talk. And then companies like Novartis, Biogen, and particularly Ionis. I've done now 3 sabbaticals in industry at Merck and Novartis, and last year at Ionis, and some of what I'll be talking about today relates to that work that Ionis has been doing. So even though they don't pay me exactly, I'm very sympathetic to what these nonprofits and companies are doing. So if I say nice things about them, I kind of keep that in the back of your mind. This is a slide that I use a lot that sort of shows the work that we do. We do here in the Neurogenetics Branch at the NIH and with a number of different diseases, and also is being done all over the world in the field of human genetics. And it's really guided our work for over 40 years now, as Luke was saying. We see patients with hereditary neurologic diseases. We characterize the clinical manifestations. We collect samples. We do DNA sequencing. Next gen sequencing, and look for variants. And then we track those variants. Look to see which specific variants are associated with the disease and aren't present in controls, and segregate with the disease in families and present with multiple families, ideally, that tell us we've got the disease gene. And we as a field have been very successful with this over the last 40 years. At last count, I just checked OMIM yesterday, there are over 6,600 human hereditary diseases with known genetic cause. That's a lot of accomplishment. Each one is really cool that you've pinpointed the exact cause of the disease. And that starts you on the bottom part of this cycle. You can look at the disease gene to characterize its normal function and see how that function is altered with mutations in the gene. Is there a gain of function or a loss of function? And you can develop cell culture and animal models that can be used to work out the disease mechanism and identify therapeutic targets. Then you can set up assays to screen for small molecules or develop biological treatments. We've been very successful, well, first in identifying all these disease genes, but we as a field have also been pretty successful at coming up with treatments that are effective in mice. I've been saying for several years now that if we had mice as patients, we'd be all set. We we could just take care of the mice, but the challenge, it's a challenge to translate results in mice, results that show efficacy, safety, tolerability in animals to treatments that are safe, effective, and affordable in patients. But we're finally making inroads in this finally able to close the loop here with a few diseases, but there are many, many more to be done where this needs to be done. So I started planning this talk. I was going to talk about 4 different diseases, but I thought with the time available, better to focus on just 2 of them, 2 different hereditary motor neuron diseases: spinal muscular atrophy, autosomal recessive disease of infancy and early childhood, and ALS, a neurodegenerative disease that comes on later in life, and when it's hereditary, it usually has an autosomal dominant pattern of inheritance. So first, SMA, for those who don't know about it, it's gotten a lot of publicity over the last few years as the treatments have become available. It's a very severe hereditary disease of infancy and childhood. It's said to be probably is the most common severe hereditary disease of infancy. And as an autosomal recessive disease, the parents are usually both carriers, and the carrier frequency is fairly high. So about 1 in 40 in people of European and Asian ancestry. So it's probably, I've lost the participant count here, but I wouldn't be surprised if at least one of us on this Zoom call is carrying this mutation and would be at risk of having children or grandchildren affected by it. The disease comes on usually very early in life, usually in the first year of life, with severe progressive weakness due to motor neuron loss and muscle denervation. Now, the gene was identified back in the mid-90s, 1996. it was an international effort that localized it on chromosome 5, and then a group in France and in Paris, Judith Melke was the lead author, identified the gene. They called it the survival motor neuron gene, or SMN. So we know that mutations of SMN are what cause the disease. And they're loss of function mutations, usually deletions, sometimes other loss of function mutations, missense or nonsense mutations. The genetics is kind of interesting in that there are two different versions of SMN, and both are present in variable copy numbers. So SMN1, which is the version of the gene which is lost in the disease, encodes a full-length protein. But every patient with the disease also has at least one or more copies of SMN2, which is nearly identical to SMN1. There's only one important sequence difference in exon 7 of the 8-exon gene that has an effect on the splicing of exon 7, so that the majority of messenger RNA transcript that's encoded by SMN2 is lacking exon 7, which is an important part of the protein. But there's a minority of transcript that's full length, that encodes a full length protein. And so the message here is that the patients have reduced but not absent levels of SMN. SMN is a critical gene, encodes a spliceosome assembly factor. It's important in every eukaryotic cell that has splicing. And so complete loss of SMN is lethal to any cell, all the way down to early embryos in yeast and in yeast. But the Pombe, at least, the patients have low levels, and that causes a selective loss of motor neurons. So it's been pretty clear for some time now that the goal here of treatment, there are other approaches to treatment, the goal is to try to up the levels, increase the levels of SMN protein. And the approach that really worked for this was with an oligonucleotide. So oligonucleotides are short stretches, usually like 15 or so nucleotides, DNA or DNA-RNA hybrids. And the company I mentioned I was on sabbatical with, Ionis, has done a great job with developing oligonucleotides targeting a range of different genes. And it was their work that led to the identification of an oligonucleotide that corrects the splicing defect in SMN2. It was work done in collaboration with an academic investigator, Adrian Craner at Cold Spring Harbor Lab. And it was a great collaboration that extended over a period of years to take the oligonucleotides from Ionis and to look to see whether they correct the splicing defect in the SMN2 messenger RNA. They did this first in cell culture with a reporter construct, and then in vivo in mice. So there are a number of mouse models that have been developed for SMA by deleting and replacing or reducing the levels of SMA. These mice come down with a kind of a mouse equivalent of the human disease. The mice, most commonly used model that's the Delta 7 mice developed by Arthur Burgess at Ohio State University. These mice become weak in the first few days of life and die from progressive paralysis within about 15 days. So in these mice, you can see, well, first in wild type mice, that there is a correction of the splice defect. So it goes in these mice from 10% exon 7 inclusion up to 90%. And with one infusion into the cerebral ventricles, you can correct it for up to 6 months, which is pretty amazing. I'm going there, sorry. And so it's long-term exon 7 retention after the infusion. And then you have this dramatic effect on the survival of these mice. So here in red, you see the untreated mice dying within 15 days. And here, the oligonucleotide given subcutaneously will prolong the survival to up to a year and more. So it really is a dramatic effect in the mice. I mean, the challenge was to get this to work in patients, and lo and behold, to make a long story short, it did. So this oligonucleotide developed by Ionis and Biogen, when given intrathecally, that's into the CSF by spinal tap, lumbar puncture, has as dramatic an effect in the patients as it does in the mice. Here's this from 4 years ago now, one of a series of trials that have been done with this agent, nusinersen, brand name now is Spinraza. These patients with the severe form of spinal muscular atrophy who would become weaker, progressively paralyzed and die within 1 or 2 years with the treatment early in life will get an improvement in muscle function here measured by the Hammersmith test. And they just keep getting stronger and stronger month by month. And if the treatment is started early enough, well, even before the onset of weakness, these kids can be normal. It's so I still get a little choked up thinking about this. It's or having worked on this disease, dealt with patients, seen patients die from this disease over the years. They can be completely normal with this treatment if it's started earlier. Now the patients who have later onset, the older patients with the disease don't get as much of a response. So it's not a cure-all for everybody, but it certainly can cure the disease when given early enough. So there's still issues to address here in terms of making the treatment available. Since nusinersen came on the market as an oligonucleotide, there are now 2 other gene-targeted treatments that are available. A gene replacement therapy, which the oligonucleotide has to be given every 4 months, but the gene replacement can be given just once. Was developed by Avexis, which is now part of Novartis. And then more recently, a small molecule developed by a biotech company called PTC and then picked up by Roche with a lot of help from the SMA Foundation. And that can be given orally daily. And that is similar benefit to the oligonucleotide. Remarkably, a small molecule that targets this splicing event can work just as well as or about as well as the oligo, and it works orally. The challenge is that each of these treatments is really expensive, and making it available to the patients who need it can be difficult. The other disease I wanted to talk about is ALS, this is also well known. Lou Gehrig's disease. It's a disease that comes on in adulthood, and it's dominantly inherited rather than recessively inherited. If it's inherited, it affects comes on in about 1 to 2 per 100,000 individuals, people. So there's a lifetime risk for us of about 1 in 900 something coming down with the disease. Usually comes on mid to late adulthood, but doesn't keep increasing with age. It peaks out in the range of 40 to 60 years of age. Men are more affected than women. It also causes progressive weakness from muscle denervation, with average survival of 3 to 5 years from the time of diagnosis. There are a couple of treatments that have been approved, riluzole and edaravone, but they don't have a big effect on the outcome. They might improve the survival a little bit, or improve lessen the decline in motor function a little bit. But a really restorative treatment like we have with SMA is not yet there with ALS. And the thing about ALS is that the large majority of patients are sporadic. They don't have any family history. They don't have an identified genetic cause. But about 10 to 15% of patients do have a genetic cause. And these patients have the same kind of clinical manifestations as the sporadic patients. So we have, as with other neurodegenerative diseases like Alzheimer's and Parkinson's disease, we have a subset of patients with defined genetic cause. And that gives some insight into the disease mechanism and the others, and also gives us a handle, an approach to treatment that's very similar to what's been used effectively for SMA. So the first gene to be identified back in '93, Bob Brown's lab when he was at Mass General, was SOD1. And there are now over 30 different genes that have been identified. Another one's more common than SOD is C9orf72 repeat expansion. SOD is what at the time of this discovery is well known function involved in oxidative stress response. There was some thinking early on that it might be a loss of function, but there was not consistent loss of function with the different mutations that were identified. And here, the large majority of mutations are missense mutations. And what these mutations do is not cause a loss of function so much as, or that's not the main problem. The main problem is a toxic gain of function by the mutant protein. Mutations make the protein more prone to aggregation, and they make it toxic to motor neurons. And you can see this in cell culture and particularly in animal models. So I think this was a mouse developed by Dave Borchelt, who's now at University of Florida. When you take a mutant version of the gene, put it into mice, they come down with the same kind of manifestations as the patient. So it's a toxic gain of function. So here the approach, also, a really viable approach, a useful approach to treatment is again with oligonucleotides, but this time not to correct a splicing defect, but to just get rid of the messenger RNA. And that's done with gapmer oligos that get into the cells, and they recruit RNase H1 to degrade the protein, degrade the messenger RNA that reduces the levels of the mutant protein. And this is very effective in mouse models, work done by a collaborative group at Ionis and Biogen. Alex McCampbell, the lead author, was a graduate student in our lab at Penn, like Al Espada. Whoops, sorry about that. Tried to inactivate that on this computer before the talk. The oligos in this mouse model, well, you see that it, again, has a marked effect on the survival of these mice, which other treatments have not. And it improves the rotor rod performance, a kind of log rolling test for the mice. And an interesting finding, which bodes well for clinical therapy, is that after the disease onset, you can see a benefit that with a reversal of the phenotype, not just a slowing of the phenotype, but a reversal of the phenotype in terms of the compound muscle action potential. So this is looking to see how many axons are in the peripheral nerve in the mouse. And you can see that after they've already begun to lose amplitude, it gets better with the treatment. And the news here lately is just published 3 months ago now in New England Journal is a phase 1/2 trial of this oligonucleotide, which they call tofersen, in patients with SOD1 mutant ALS. And this is a multiple ascending dose study. It was sponsored by, as a drug developed by Ionis and sponsored by Biogen, at 50 patients at 18 sites in the US and Canada and Europe. It was a short-term study, 4 different doses and placebo given intrathecally at 2 to 4 week intervals. And the patients were followed for a relatively short period of time, just 12 weeks of intervention, 12 weeks of follow-up. They found that the treatment was well tolerated. The patients had some issues related to getting the spinal taps to deliver it, pain at the injection site, post-LP syndromes. But overall, it was well tolerated. And the main finding is that they were able to reduce the CSF levels of SOD1. Here are the 4 different doses. You see the lowest dose that didn't have any detectable effect, but all 3 of the higher doses led to a reduction in the levels of the protein to 25 to 33%, which is equivalent to the level that was reduced in the mice to give the clinical benefit. So the idea here is if you can follow these patients longer, look at a larger number of patients, you might see clinical benefit. In this 12-week study, they did not see a clinical benefit overall, but they had set up the study to look specifically at patients who have mutations that are known to cause a rapid progression. There's one particular missense mutation, A4V, it's common in North America, that causes a rapid downhill course in these patients. And looking at the ALS functional rating scale, you can see a significant difference between the patients on placebo and those in the higher dose range. And likewise with the breathing capacity and with the grip strength, the handheld dynamometry. So just the conclusions here. In conclusion, the patients with ALS caused by SOD1 mutations, CSF concentrations decreased with the higher doses given intrathecally over 12 weeks. Some adverse effects, but overall the tolerability was good. And the evidence of target engagement that's showing that they could reduce the levels of the toxic protein and the tolerability, and some hints that it could be effective, at least in the more severely affected patients, really bode well for the future. And now there is a phase 3 trial, larger study with 99 subjects at 36 sites, 19 of them in the US, looking at the higher dose with the primary outcome measure being the functional rating scale, following patients for a longer period of time, 28 weeks, and that's due to be finished by within less than a year, and we'll see whether it works. The point here is that the same kind of approach can work for a number of different diseases. There are clinical trials now underway with oligonucleotide treatment for other dominantly inherited diseases with toxic gain of function, particularly Huntington's disease, which is, maybe about a year ahead of ALS with SOD, and Parkinson's disease, LRRK2 and alpha-synuclein, tau and frontotemporal dementia, and other familial forms of ALS, including C9, are in clinical trials. And there's work to develop similar kind of treatment for other polyglutamine expansion diseases like Huntington's. So, the editorial that came out with this article in the New England Journal from Orla Hardeman and Leonard Vandenberg said that this is the beginning of genomic therapies for ALS. I would suggest that we're really at the very beginnings of genomic therapies for lots of these diseases. Nearly all of these 6,600 diseases could be targets for this kind of treatment, oligonucleotides, gene replacement, gene correction, small molecules. And that's for the rest of you guys to pick up and run with if you're interested. We could use a few good people to take this on. And, the point, theme here is that it takes, it's a collaborative effort, collaboration between disease foundations, the NIH, academic medical centers like Duke, and industry that gets this to work. Maybe just close with a quote I like to use, I've been using lately, by philosopher Reinhold Niebuhr, that nothing we do, however virtuous, can be accomplished alone. This is a picture from, we didn't do it this year because of the virus, but this is our branch outing from a couple of years ago, showing some of the members of the branch. This isn't everybody in the branch, and there are a few people in here that aren't actually members, that they don't work in the lab, they're too young for that, but we basically got everybody together in the park for this group photo. And it's a great place to be here, and welcome people want to come here to do part of their training, take jobs. It's a really good place to be, and we try to put the government's money to good use. Also, just a folk, Audrey didn't make the group photo, but there's a graduate, or just got her PhD, now a postdoc in our lab, Audrey Winkelsauce, who's affected by SMA and has been working on her disease to come up with supplemental treatment, other kinds of approaches to increasing SMN levels that might work in older patients, work better in older patients like her. And so, it takes a village, and it's a collaborative effort that makes this happen. So questions? I don't know if I ran over too much, Lou.
No, that was perfect. Thanks for that talk. That was really, really great. Yeah, we're going to go into a question and answer session. So if anyone has a question, feel free to either drop it in the chat or just
wow. It's good to see Aldo Spada's joined in. I hope I didn't say anything bad about Aldo.
Yeah, well, that was just before we started.
That's right.
Okay, well, I'll start off with a first question. These are like really exciting results to see for these genetic diseases. And do you think there's anything that we can learn from or apply from these ASO trials to either like, well, specifically for ALS, like the sporadic patients, or some of these other like more complex neurodegenerative diseases?
Yeah, well, that's why, others have seen the challenges you asked for. So number one challenge is identifying therapeutic targets. And I think identifying a target is pretty straightforward, or at least some of the best targets are the disease genes themselves when you have one. So in the diseases I talked about, SMA and ALS, you know the cause of the disease. And the same thing holds for other diseases like Huntington's and the forms of other hereditary forms of ALS like C9 repeat expansion. Targeting the disease gene itself is it is enough. Well, it works. We can see that it works here, and at least for SMA and probably for others. The challenges with the sporadic diseases, as you say, is to identify therapeutic targets, and so that means getting to know something about the disease mechanism. So with ALS, we've come to learn a lot over the last 5-10 years about the disease mechanism, TDP-43, accumulation in the cytoplasm, loss of TDP-43 function in the nucleus, the nuclear cytoplasmic transport, mitochondrial function. So, you can target all these different aspects. So for example, TDP, with the TDP-43 mechanism, there are downstream targets that plays a role in, also in splicing and intron retention. And one downstream target is Stathmin-2 that can be targeted. So, maybe you can correct the splicing there, like in SMA. Maybe you could work, some of the other targets that are implicated in sporadic forms of the disease. Same thing with Alzheimer's and Parkinson's, and prion diseases. It's more of a challenge, but good use of the cell culture and animal models points towards therapeutic targets. There's a long answer to a short question.
That's great. It's a complicated question. Anyone else? Questions? I have more.
Oh, there's there are a few in the chat. Let's see.
How do you want to unmute and ask, or you want me to ask it for you?
Right. I can see it here. Al asks, Dr. Laspada asks, can I comment on, here we go, how much expect tofersen to cost? Seems like a recurring issue is that these therapies are prohibitively expensive for many. Okay, well that was the third of my challenges, right? The first one was identifying therapeutic targets. The second one, I could say something about it, because it's a challenge sometimes, is how to translate from mouse results to human. And then the third is the important one that Al's calling attention to here is the cost or availability. So SMA has been a good experience there. I mean, or a bad experience, I might say. It shows us what comes up. So the price tag, which I think was kind of arbitrarily put on nusinersen, is about $125,000 per treatment. It's given every 4 months. There are newer versions of it that maybe be given less often, every 6 months, but still, it's expensive. And several of us gave them a hard time when they came up with that price, when Biogen came up with that price. But it's what they could charge and what they could get away with and what they could get insurance companies to pay for. And it does cover their expenses. It helps them to develop other newer drugs. And then, as it turned out, when the gene replacement came along, Zolgensma, they let's see, that was Avexis at the time, and now Novartis. They charge. It's a one-time treatment. They charge over $2 million, $2.2 million, I think. And then more recently with Risdiplam, where it's given orally, they kind of peg their price based on the others, and so it's like $1,000 a day. And you know, so once one gets away with charging that much, then the others come along. So yeah, I think tofersen is going to be expensive if it works in this phase 3 trial and gets approval. And I think that's something we have to address. I think it's one of those challenges that's out there. And there's a lot of interest, you know, bipartisan interest here in Washington to try to do something about the high cost of drugs in general. But this is a particularly egregious example. There was a CNN special last week about an SMA patient in Canada whose parents had to raise, or they did raise $2 million for the baby to get gene replacement therapy for spinal muscular atrophy. And they did a kind of GoFundMe thing where they were able to raise that much money. But Novartis has this deal where they have a lottery where you can get it for free. Biogen likewise for nusinersen, they'll make it available if your insurance doesn't pay, which is about a quarter of the patients that they give it to. So, you know, the companies do have, the people at the companies do have a heart. You can work on that some, but I think it's going to take some action at state level, national level to try to put a lid on it, because it's not a competitive market. It's a, you know, whatever they get away with. Al, do you have something to add about that, or your perspective?
Well, I mean, I wonder if, you know, this is sort of, you know, if they're going to shoot themselves in the foot with this approach, right, and that they're going to definitely sort of pave the way for, you know, for companies to pursue one and done therapies, mm-hmm, where, you know, there's a single administration that provides, you know, long lasting, you know, therapeutic success, you know, and at least with that, you know, it's just a single cost. And because you can imagine SMA, if SMA patients end up living a normal lifespan and they need this drug for, you know, 50, 60, I mean, it's a little mind boggling to me, you know, how this could be a, you know, a sustainable, you know, neurotherapeutics resolution, you know, for these diseases that we're so interested in and so passionate about. I don't know. Anyway, it's a good problem to have, right? You know, to have treatments that, you know, that are so successful and could even, you know, you could even use the term cure on rare occasion, maybe with SMA when you treat patients before they develop symptoms. I just don't know. You're in Washington, so I figured if anyone, well, Bethesda, close enough. You know, this is the sort of thing that you know, it seems like NIH should take on at some point.
There was talk about it, you know, because a lot of the, for each of these diseases, a lot of the early work is funded by NIH, that the NIH should have margin rights to you know, put a lid on the pricing. But that didn't fly. Francis Collins didn't want to go there. You know, the goal here is to develop treatments and then let somebody else worry about how much they cost. But yeah, I think it's important. And I think it's going to have to come from outside the industry. The risk is, as I was suggesting, is that it's going to break the bank. You know, for national health plans and for insurance plans, insurance companies, that they just, as the N increases, as the number of diseases where we have this kind of treatment increases, we can't keep it up at this rate. And I think the company's approach is just to make as much money as they can while they can. They owe it to their investors, I guess. It's going to have some kind of limit on it. You guys can work through your elected representatives to try to do something about this too, I think, or to support it. I think. I guess I'm not supposed to get too political in these things, but.
That's okay. You can't get in trouble, Kurt. You're beyond reproach at this stage.
Yeah, right. It's but it is a challenge. The other aspect of access is, you know, for SMA, and I think it's going to be true for the others like ALS and Alzheimer's and Parkinson's, is how do you identify patients at a point where they can benefit early on? And for SMA, that means doing newborn screening, and that's catching on. Last I heard, I think it's probably more than that now. There are at least 12 states that have newborn screening for SMA to start patients on treatment before they become symptomatic is really important. And I think, you know, for ALS, one finding that makes that, you know, that I think is kind of intriguing is the increase in neurofilament levels before onset in patients with hereditary forms of the disease. So that would be a way to identify people before they become symptomatic and start the treatment early. Price and starting treatment earlier are two factors that need to be addressed, kind of socio-politically, I guess, as well as biologically, biomedically.
Right. Yeah, these are super important issues that hopefully that we as a field can tackle moving forward. And I think it makes a really good segue into our group discussion period that we're going to go into. And so I'm going to get us ready for that. And so for these group discussions, we have 3 questions that I've kind of formed out of the 3 major challenges that Kurt was talking about. And so in our groups, hopefully we can work through these. How can academic researchers most effectively translate their efforts towards patient impact? How can we involve more diverse collaborations to navigate research barriers? And how can we ensure new therapies are widely available to all and effective for all who need access to them? And so Pia is going to help put us into breakout rooms so that we can kind of dive into these on a bit of a deeper level. Marg had a really good question in the chat that that maybe we can take that into the breakout group as well. But yeah, for those who are interested and willing, let's dive in.